In class, we constructed cladograms, and there are some examples in the online quizzes for you to practice. In general, I will give you a couple of groups and a couple of features, and I will provide a table in which the organisms are depicted as either having a trait, in which case they have a 1, or lacking a trait, in which case they have a 0. And from this, using the principle of parsimony, you should be able to construct a cladogram. So any group, therefore, which has all zeros, which doesn't have any of the derived synapomorphies, they would then be the outgroup and the first branch from the family tree. If there is an organism which possesses one of the features, but only one and has zeros in the remaining traits, this then would be the second branch from the family tree. The third branch would be the organism which possesses two number ones, the remainder are zeros, and so forth. The very last branch of the family tree would be the organism which possesses ones for each of the categories. So on the exam, if I give you a chart with this information, you should be able to draw a cladogram as are represented in the online practice exams. As you review the question sets, and especially as you go through the genetic evidence question set in the Evidence of Evolution folder, you should become familiar with a number of aspects of genome comparisons. So, for example, human and chimp genomes are incredibly alike. We are 97% chimp or so, genetically speaking, and chimps are 97% human, give or take, genetically speaking. That chromosomal changes underlie many evolutionary changes, so that there are inversions and translocations, etc., which can then be linked to different groups. There was a mouse-human chromosome comparison that you were provided with, and that two small chromosomes fused to make one, as the 48 chromosomes in the ape genome was modified to become the 46 chromosomes in the human genome. And not only can we look at the existence of genes and the sequences of genes which code for proteins as being significant in evolution, but also what we call non-coding DNA. So many sections of DNA do not code for protein, but code for RNA, but their modifications not only show evolutionary patterns, which we can use to study evolution, but were also very important in mediating evolutionary change, including jumping genes. In fact, there is one group of jumping genes known as ALU sequences, which have been especially important in primates and the higher primates such as ourselves.